Hello student, in this class I will discuss the conclusion behind experiment 4. In the last experiment or experiment 4, the scientist was taken heat kill H3 with RII are injected into mice and observed that the mice was dead. Why mice was dead is very important because we know that when the scientists taken individually heat kill H3 injected into mice and observed that the mice was survive in the experiment 3, but when the scientists are taken RII type are injected into the mice and observed that the mice was survive in the experiment 2, but when the scientists are taken both the thing heat killers III with RII are injected into the mice and observed that the mice was dead. Mice was dead. Why mice was dead? The mice was dead due to recombination of gene. The mice was dead due to recombination of a gene. That means in the last experiment, the scientists are taken heat kill III with RII are injected into mice and observed that the mice was dead. The mice was dead due to recombination. The mice was dead due to recombination of a gene. Then what is the recombination of a gene? The recombination of gene is nothing but the recombination of a gene means when the scientists provide heat to this virulent type bacteria. That means suppose I can first draw the structure of bacteria. Suppose these are the virulent type bacteria and these are the avirulent type bacteria. The virulent type, what is the virulent type? We know that virulent means those microorganisms which is responsible creating disease inside the living system known as virulent. Avirulent, those microorganisms which is not responsible not creating disease inside the living system known as avirulent. And the virulent create a capsule layer, that means a outermost covering. Suppose these are the bacteria and this bacteria surrounding a capsule layer because these are known as virulent, and inside this bacteria we are found the genetic material or genome or a DNA. And in the avirulent type bacteria the outermost capsule is absent, so it is called avirulent or rough cell bacterium, otherwise known as rough cell bacterium. And inside this bacterium we are found the genome. Recombination of a gene means when the scientists provide heat to this virulent type bacterium. That means when the scientists provide the heat to this virulent type bacterium or S triple type bacterium, the capsule layer is degenerate, but the disease causing gene are present inside this virulent type bacterium. This gene enter into the avirulent type. This gene enter into the avirulent type or R type bacterium and recombine with R type or avirulent type bacterium, converted the avirulent to virulent form, so mice was dead. Is clear?